This section explains why nanotechnology sits at the root of regenerative engineering. The seed for the whole book is one observation. Cells do not only read molecules, they read shape at the scale of their own machinery. That observation is what sets nanotechnology apart from chemistry. The discipline attends to the physical structures that present chemical functions rather than to the chemistry itself. Geometry becomes the signal. Fabricate features in the band from 1 nanometer to less than 1,000 nanometers, and you reproduce the instructive microenvironment that tells seeded cells to reassemble into tissue. The authors deliberately stretch past the textbook range of 1 to 100 nanometers because the cues a cell actually senses, the fibers of the extracellular matrix, the growth factors, the organelles, live in the nanometer to micrometer range. This is why the field renames itself regenerative engineering. The aim is to supply the form from which tissue regenerates itself. This section explains the pivot that organizes the chapter on the body. The move is simple to state. Stop building the organ. Supply the boundary conditions and let the cells build it. That is the chapter's turn, away from biomaterials as inert replacements. The body walls off, the foreign body response with its imperfect integration towards scaffolds that instruct seeded cells to spontaneously reassemble into tissue, then resorb as those cells lay down their own matrix. Regeneration becomes a self-assembly problem. Specify the niche and the self-assembly problem. Specify the niche and the cells solve the rest. The constraint the whole approach hinges on is scale. Cells read their surroundings in the nanometer to micrometer range because the native ECM is a nanostructured web of collagen, laminin, and GAGs. So manipulating matter at 1 to 1,000 nanometers is the lever on cellular fate. Chemistry alone never reached this register. Chemistry alone never reached this register. Chemistry alone never reached this register? This section explains how a cell reads the shape of its scaffold as a direct instruction. A cell reads its substrate's geometry as instruction. Native tissue runs on a fibrous extracellular matrix built from collagen, elastin, fibronectin, and laminin. The chapter's claim is that copying only the form, nanofibers at the 10 to 300 nanometer scale of real collagen fibrils already transmits the signal before any biochemistry is added. The fiber couples to the cell through integrin and actin linkages, so topology propagates inward to the cytoskeleton and modulates gene expression. One geometric variable thereby steers the entire behavioral repertoire. The chapter then unfolds: polarity, adhesion, proliferation, migration, orientation, and differentiation. The scaffold is not inert support. It is an extracellular matrix analog whose shape is the message, and the design knob is fiber architecture, not coding. This section explains how a cell reads the physical shape of its surroundings, not only their chemistry. A cell senses the geometry of where it sits. The native extracellular matrix is a mesh of nanofibers, collagen, elastin, fibronectin, laminin, fibrils 10 to 300 nanometers wide. The cell couples to that geometry through integrin and actin linkages, which let fiber diameter, alignment, and curvature dictate its polarity, alignment, and curvature dictate its polarity, adhesion, migration, orientation. The chapter's move follows from this. If geometry is itself a signal, then an engineered scaffold need not deliver growth factors to steer cell fate. It need only reproduce the nanofiber form, recruiting the cell's own mechanosensing to do the directing. Tissue engineering then becomes a problem of building the right physical channel. the analog of the extracellular matrix, rather than dosing the right molecules. This section explains why regrowing bone past a critical defect needs a built template, and what that template is for. Bone will not regrow itself past a critical defect, so you build it a template. The template's job is to wear bone's architecture. It carries the shape of bone rather than the substance of bone. Native bone, extracellular matrix, is a felt of collagen nanofibers 50 to 500 nanometers across. Cells read that geometry as instruction. They differentiate toward osteoblasts even without chemical coaxing. The chapter's whole move is this: copy the shape of the matrix at the nanometer scale, and the resident stem cells do the building. Electrospinning is how you write that shape. An electric field draws a polymer jet into fibers of tunable diameter. Everything downstream, the three-dimensional bulking, the polymer choice, the ceramic reinforcement, is making one flat mat closer to the real tissue's form. This section explains why bone regeneration is a copying problem. Regeneration is a copying problem. Bone heals when it is handed a scaffold whose geometry the cell already knows how to read, so the work is faithful, reproduction of a structure the body recognizes, rather than fresh construction. The reason sits in the matrix. The native extracellular matrix, the ECM, is a composite of two ingredients: collagen nanofibers ranging from 50 to 500 nanometers supply resilience, and mineral supplies stiffness. A cell that lands on fibers at that exact scale behaves as if it were home. So the whole engineering task collapses to one thing: manufacture fibers at the ECM's length scale. Electrospinning is that tool. An electric field overcomes the surface tension of a polymer droplet, drawing out a jet that dries mid-flight into nanofibers. Everything downstream in the chapter serves a single fidelity. The fabrication tricks, the polymer choices, and the ceramic reinforcement all bend toward one goal: reproducing a matrix the body already recognizes. This part explains why bone grafting and bone tissue engineering exist as a field at all. Bone already knows how to rebuild itself scarlessly. Surgery only buys structure and time while it does that. So the graft's entire job is a conjunction. It must bear load and let cells migrate in and induce new bone and grow its own blood supply all at once. The field exists only because those demands are mutually antagonistic. Consider how each material fails. Dense ceramic wins strength but won't osseointegrate. An open porous scaffold invites cells in but buckles under load. Autograft is biologically perfect, but you cannot harvest enough of it without maiming the donor site. Every named option maxes one axis and forfeits another. Tissue engineering is the wager that simultaneity itself is buildable, that the prize lies in satisfying all of these demands together, rather than in maximizing any single virtue. Bone is the hardest scaffold problem in the book, and this section explains why. The reason is a contradiction. The tissue forces onto any replacement for it. Stiffness comes from density. Regeneration comes from porosity. Vasculature, cell traffic, and remodeling all need void space, while the 10 to 20 gigapascal modulus of cortical bone needs the opposite of void. The whole chapter is one move repeated: graded stiffness, growth factor, angiogenesis, and biomimetic mineralization each try to buy both ends of that trade-off at once, rather than concede one of them. Bone is the book's hardest instance because its two demands sit most directly opposed. Read it as the template that the softer tissue chapters relax. The scaffold is a temporary contradiction. It is held just long enough for the body to dissolve it and regrow the real thing. This part explains why cartilage needs a scaffold and what a scaffold has to be. Cartilage cannot repair itself because it is avascular. There is no blood supply, and so no healers get recruited to the wound. The only route is to build the missing tissue from outside. The chapter's primitive is this. A scaffold is defined entirely by what it must reproduce of the native tissue it replaces. Every design constraint is back-translated from one native feature. Interconnected pores echo the lacunae the cells live in. Multiphasic mechanics echo the water-driven cushioning. Surface charge and roughness echo the cues chondrocytes read to stay chondrocytes. A scaffold's telos is to make seeded cells behave as if they were already inside cartilage, and then to degrade as they rebuild the real thing. This part explains why cartilage cannot repair itself and what that forces every tissue engineer to do instead. Cartilage cannot heal itself because it is avascular. There is no blood supply, no route for repair cells to reach the damage. That single lack defines the entire engineering problem. You cannot deliver healing. You can only stage it. The chapter's primitive is that a scaffold never builds the tissue. It imposes the boundary conditions, pore geometry near 150 to 250 micrometers, compressive stiffness, surface charge, and a degradation rate clocked to matrix deposition, whose only stable outcome is native cartilage. The chondrocytes do the building. The scaffold merely selects what they build. Everything downstream. Natural versus synthetic polymers, biomimetic modification, is searched through the space of constraints for the set, whose fixed point is hyaline cartilage. This section explains why skin tissue engineering, despite decades of progress, has not yet built a whole skin organ. Skin tissue engineering follows the same compositional recipe. The book repeats: scaffold plus cells plus chemical and mechanical stimuli. The skin instance is valuable because it exposes which of those terms is the binding constraint. Rebuilding the barrier itself, keratinocytes grown on a collagen-like matrix, was solved decades ago, and commercial substitutes already exist. What no construct delivers is the whole organ, pigment, appendages, and above all, perfusion. Past a few hundred microns, diffusion starves the cells, so a thick graft survives only once. Blood vessels grow into it. The chapter names this the conundrum of vascularization. So, this chapter is a progress report. Every advance it covers, new marine polymers, growth factor delivery, bioprinting, skin on a chip, is scored against one target that remains unbuilt, a fully vascularized appendage-bearing skin that regenerates without scar. This section explains the body chapter, where skin is the book's standing recipe. A scaffold seeds, cells, chemical and mechanical cues steer them, a stratified barrier reassembles, and the whole thing runs on the largest organ. What shifts per chapter is which variable binds. For skin, the binding constraint is vascularization. A construct thicker than diffusion can feed, roughly 200 micrometers, starves at its core unless capillaries grow in fast enough. So the real target is the perfused dermis beneath the surface, since the epidermal sheet already grafts well on its own. Every later move answers that one bottleneck: pro-angiogenic factors, bioprinting, even photosynthetically delivered oxygen. Read the chapter as the same scaffold, cells, and signals triad, with feeding the tissue promoted from an afterthought to the whole problem. This section explains why neural regeneration scaffolds take the shape they do. The pattern is simple. 2 nervous systems, 2 failure modes, and the scaffold's form is just the shadow of which one you face. In the peripheral nervous system, the PNS, an axon already wants to regrow. The only obstacle is a gap with no path across it, so the engineering is guidance. You build a solid nerve guidance conduit that bridges the 2 stumps, an engineered stand-in for the autograft. In the central nervous system, the CNS, the gap is not the problem. Here, the environment is actively hostile. Glial scar, inflammation, an impassable barrier. So the engineering is suppression and delivery. You pour an injectable hydrogel into the lesion to ferry cells and drugs. The whole chapter is that one bifurcation. Match the scaffold form, a liquid field, versus a solid channel, to the geometry of the failure. This section explains the single asymmetry that organizes the entire chapter on nerve regeneration. One fact governs every material choice. The PNS regenerates itself. The CNS does not. Every choice of biomaterial falls out of that one fact, where the axon already wants to grow back. In peripheral nerve, the scaffold's only job is guidance. That gives you solid nerve guidance conduits, which channel regrowth across a gap. Where the tissue has surrendered its own capacity, in the brain and spinal cord, the scaffold must substitute for that capacity while defeating 3 active suppressors. The first is the blood-brain barrier, which locks drugs out. The second is inflammation. The third is the glial scar, which walls off the wound. That gives you injectable hydrogels, ferrying cells and growth factors into hostile terrain. Same biomaterials toolkit, 2 opposite mandates, selected entirely by how much the body still does on its own. This section explains why the eye is tissue engineering's cleanest proving ground, and how the chapter is built. The chapter is organized by walking the light path: cornea, conjunctiva, lacrimal gland, lens, vitreous, retina, optic nerve, engineering each a vascular part on its own. The structural gift is immune privilege. A vascular tissue feeds by diffusion, so a foreign scaffold draws little rejection, widening the menu of usable materials and cells, and the simplest structures are just stacked sheets, easy to mimic. The surprise is that this freedom buys nothing where it counts. Ophthalmology lags skin and bone because vision demands one construct that is simultaneously biocompatible, optically transparent, and mechanically strong. A triple bind no other tissue imposes. Privilege relaxes one constraint. Transparency reimposes a harder one. This section explains why the eye chapter works as the controlled experiment of an organ-by-organ book. The eye is where you run tissue engineering with 2 of the field's hardest constraints switched off. The cornea, lens, retina, and vitreous are avascular, fed by diffusion, so there is no vascular network to reconstruct. They are also immune privileged, so graft rejection relaxes and the palette of materials and cells widens. What remains when those 2 drop away is the constraint no other chapter faces. The construct must be optically transparent while still biocompatible and mechanically sound. That is the chapter's primitive transparency as the orthogonal axis. This explains the paradox. The introduction states flatly, the eye is the simplest tissue environment, yet ophthalmology's clinical progress is the slowest, because the eye trades familiar constraints for one the field has no template for. This section explains why restoring a blood vessel is a problem of rebuilding structure, not of fabricating a tube. A blood vessel is a living three-layered control surface. The endothelial monolayer lining the lumen is what makes it thromboresistant, selectively permeable, and tone-regulating. An inert tube that lacks this lining, such as Dacron or EPTFE, clots shut below about 6 millimeters in diameter. That failure is the chapter's whole argument. Vascular function is structural, so restoration means recapitulating the intima, media, and adventitia organization that generates the behavior. Extruding a conduit that merely matches the bore does not restore the vessel. Every strategy surveyed answers the same question: how to grow the hierarchy back. These strategies are decellularized matrix, scaffold, seeded cells, and scaffold-free cell sheets. The failure of the small diameter graft is the proof that geometry alone was never the vessel. This section explains why a blood vessel defeats the tissue engineer, and how reading the vessel by layers turns that defeat into a plan. The structural surprise is that the vessel matters as a lining before it matters as a tube. Subramanian's chapter 9, sets the target. A synthetic graft holds pressure perfectly well, yet the small diameter ones, smaller than 6 millimeters, clot and occlude because they lack the endothelial monolayer that makes the native lumen resist thrombosis. The failure is biological rather than mechanical. The fix restores the living cell layer, not the geometry. The whole chapter follows from one reading. Take the vessel's three-layered hierarchy, the intima, the media, and the adventitia, and read it as a function per layer. Then put the same question to each tissue engineering route, the decellularized scaffold, the biomaterial conduit, and the scaffold-free cell sheet. The question is how each one reinstates that living interface. This is the book's recurring move. Name the tissue's hierarchy, locate the failing layer, and engineer toward it. This section explains the bio-artificial pancreas at its structural level. Living beta cells fail for a reason worth stating precisely. It is not that they cannot sense glucose and secrete insulin. Transplanted islets do exactly that. They fail because the immune system destroys anything foreign. The chapter's thesis is a single move: substitute a semi-permeable membrane for immunological self-tolerance. Wrap the islets in a microcapsule made of alginate roughly 200 to 1,000 micrometers across, tuned so that small molecules cross freely. Glucose diffuses in, insulin diffuses out. While antibodies in immune cells are too large to fuse in, insulin diffuses out. While antibodies in immune cells are too large to pass. The graft keeps its glucose in insulin out feedback loop, yet becomes invisible to rejection. This dissolves both barriers to islet transplantation at once. No immunosuppression is needed, and xenogenate donors become usable. A pancreas replacement reduces to a diffusion, selective shell around cells that already work. This part explains diabetes as a control problem, and the trick a capsule device uses to fix it. Diabetes is a broken controller. The islet feedback loop that senses glucose and titrates insulin is destroyed or ignored. Transplant a working controller, meaning donor islets, and 2 gates close it off. The host immune system reads the graft as non-self, and donors are scarce. The chapter's move is to decouple function from identity. Wrap the islets in a semi-permeable capsule, sized to pass glucose in and insulin out while blocking antibodies and immune cells. Selective permeability substitutes for immune tolerance, a size filter standing in where you would otherwise need immunosuppression or self and non-self matching. The whole device is that one trade, a membrane that grants metabolic access while denying immunological recognition. This section explains why hepatic tissue engineering exists and how the whole chapter is organized. Donor scarcity is the forcing function. Orthotopic transplant works with roughly 86% one-year survival, yet about 1,500 waitlisted patients die every year in the United States alone. So the field builds substitutes. The spine of the chapter is that the liver resists substitution because it is defined by a function set, cytochrome P450, detoxification. Fetal hepatocytes proliferate yet cannot clear ammonia, and protein and lipid metabolism, all carried out across a precise multicellular architecture. Every shortcut buys one property by surrendering another. Immortalized cell lines give unlimited expansion but lose metabolism. So the survey reads as a ladder of escalating fidelity. The rungs run from isolated cells to support devices, to biomimetic scaffolds, to recellularized, decellularized matrices, to bioprinting, and finally to organ bud transplant. Each rung recovers more of the function set, and none yet restores its long-term whole. This section explains the single principle that orders the entire chapter on liver therapies. Every therapy is ranked by how much of the liver's emergent architecture it reconstructs. Hepatic function is not one output, but a composite, metabolism, detoxification, synthesis, arising only from the right cells, held in the right cells, held in the right cells, held in the right matrix, with vasculature. Whole organ transplant sits at the ceiling, and donor scarcity is its only flaw. Below it, the strategies climb that same ladder: infused cells, extracorporeal support devices, engineered three-dimensional matrices, recellularized, decellularized scaffolds, bioprinting, and organ bud organogenesis. Each one recovers more of the native cell, matrix, and vascular organogenesis than the last. Read every section as one rung on that ladder. To predict the next instance, remember that any new method's worth is just its architectural fidelity. This section explains the core substitution behind laser-assisted bioprinting for tissue engineering. Every other way to print cells pushes them through an orifice. Inkjet extrusion, the orifice is the tyrant. It clogs, it shears living cells to death, it floors your resolution. Laser-assisted bioprinting deletes the nozzle. A pulsed laser hits an absorbing film under a thin layer of cell-laden bio-ink. The flash vaporizes a pocket of it. A bubble swells and collapses. The Rayleigh Plesset dynamics, and the recoil throws a single jet that breaks into droplets onto the receiver below. No aperture means no clogging, no shear. So you can print viscous ink at high cell density and near single-cell resolution. The chapter's whole arc is this one substitution, energy, where the whole used to be. This section explains the one fabrication method in the book that places ink with light instead of pushing it through a hole. Every other method pushes ink through a hole. This one throws it with light. A laser pulse strikes an absorbing film behind a thin layer of liquid bio-ink. The pulse flashes a cavitation bubble, and the collapse of that bubble spits out a single jet, cells and all, onto the substrate facing it. This is the lift primitive covered in Chapter 12. Because there is no orifice, there is no shear, and no clogging, so living cells survive at near single-cell resolution. The whole chapter is one mechanism viewed through many materials. The same bubble-to-jet impulse deposits metals, proteins, DNA, hydrogels, and endothelial cells. That fixes the boundary. Anything you can spread as a film, light can throw. Anything you can only feed as a stream still needs a nozzle. This section explains tissue engineered medical products as a single structural idea. Replace the donated part with the part that teaches the body to grow its own, then vanishes. That is the whole chapter. An autograft or allograft is the tissue itself, paying in donor site morbidity or immune rejection. A tissue-engineered medical product is instead a sacrificial template. It is a biodegradable scaffold mimicking the extracellular matrix, sometimes seeded with the patient's own cells or growth factors, engineered to support adhesion, proliferation, and differentiation, and then to dissolve, replaced over time by native tissue. One pattern, 6 instantiations: bone, cartilage, tendon, tissue interface, skin, and nerve. The scaffold's material and architecture are retuned to each tissue's mechanics, while the recruit then degrade logic stays invariant across all of them. This section explains the single design move shared by every tissue-engineered medical product. Every tissue-engineered medical product runs one move across 6 substrates. Build a temporary scaffold. The body reads as native. Let cells colonize it. Then degrade exactly as fast as real tissue fills in. Success is measured by the implant's own disappearance. The scaffold is an erasable template, the path the tissue regrows along rather than the tissue itself. From bone to cartilage to tendon to interface to skin to nerve, only the blueprint copied changes. Each native architecture, the hydroxyapatite lattice, the collagen mesh, the aligned fascicle, dictates its synthetic echo. The chapter catalogs that single pattern per tissue. The target structure fixes the graft's design, and biodegradation becomes the design goal itself. This section explains why the toxicity of a nanomaterial is a property of its physical state rather than its chemistry. The chemistry alone does not fix the hazard. The same carbon is inert as graphite, yet cytotoxic as a high aspect ratio. Nanotube because size, shape, surface area, concentration, and agglomeration together set how the particle meets the cell area, concentration, and agglomeration together. That meeting is what you can change. A nanotube settling and clumping on a membrane starves it. Functionalize the surface or dilute it in a polymer matrix, and the toxicity recedes. So safety is tunable. The generative primitive is this. The hazard lives in a high-dimensional descriptor space, not in a material identity. You cannot read danger off the name of the substance, only off where it sits in that space of physical descriptors. The challenge ahead follows directly. No single assay can certify a nanomaterial because the material is never one thing. Each physical form is a separate object to test. This part explains why the very smallness that gives nanomedicine its power is also where its danger lives. The same smallness that buys the function carries the hazard. Every prior chapter exploited the nanoscale, the high surface to mass, cratch, brush, while C to mass ratio, the high surface to mass ratio, the size-dependent physiochemistry to win therapeutic powers. The bulk material cannot reach, barrier crossing, terignosis. This closing chapter names the bill. Those very novel properties void the parent materials. Safety becomes a coordinate. Read off 2 axes. The first axis is the particle's intrinsic properties, size, shape, surface, composition, and degradation products. The second axis is the host's handling of it, the classical ADME path, where it enters, where it sequesters, how it is metabolized, and how it is excreted. Function and risk are the same. Physics